So our project is developing a novel heuristic to classify structural differences between DNA ligase isoforms. So it's this way.
Yeah, right. So starting off the abstract, the Euclidean of the structural function of the protein sequences is a long-term challenge in information-based structural biology. We seek to develop a heuristic using machine learning concepts to identify a protein in a completely automated process using a k-nearest neighbor classifier, which works based on the assumption that similar points can be found near one another. We utilize the information of the defining features of a protein to map out the orientations and translations within DNA ligase that define its structure as a means for identification. Compared to previous methods, we suggest a novel and geometric approach towards protein classification and structural prediction. With the rise of computational power and increasing costs in physical chemistry, it provides an area for further investigation into an easier means of protein classification. Not further introduction. There are a lot of resources required for the viability of physical synthesis of protein biologics. To be able to successfully synthesize a protein, we need to get its crystal structure, which can take up years on end, coupled with a lot of financial barriers. This is without factoring in the possibility of manual error that can occur during this time-consuming process of manual synthesis. In contrast, using a novel machine learning approach has the potential to save time, money, and promises to improve efficiency without the barrier of manual error. To be able to successfully classify a protein, we need to develop a heuristic that can act as a structural identifier for the protein. For this, we use relative geometry of some important features in a protein, such as binding sites, active sites, the presence of DNA, etc. By developing a heuristic, we can efficiently classify proteins based on their relations to one another and can eventually be used for further testing of the biological molecules and structural prediction.
Our heuristic is dependent on the relative geometry of the DNA ligase molecule, depending on the center of the DNA molecule that has been integrated. So this allows us to measure the relative distance of various factors, such as the factors Sakat stated in the introduction, such as the active site, binding site, alpha helices, and etc. Through this, we collected a data set of values so that we can run our machine learning algorithm on it. So we're going to run a k-nearest neighbors classifier in order to classify the type of DNA ligase based on the distances between the certain features. So in this figure here, this pink box is highlighting the active site and the alignment of DNA ligase 1 and 4. So these human DNA ligases have 3 different isoforms: DNA ligase 1, 3, and 4. So that'll be used throughout this presentation. So you can see the DNA molecule in orange and blue, which is this double helix strand here, and the ligands of DNA ligase 4. By measuring the distances to molecules such as these from the center of the DNA, we can gain a variety of factors that we can use to classify DNA ligase through the relative geometry. In order to determine the heuristic, we went through a series of steps in order to find the differentiating factors within models for DNA ligase. Using the sequence alignment tool on the PDB and PyMol, we determined the following factors as being important towards a model: active sites, binding sites, ligands, alpha helices, and beta sheets. These 5 primary components of DNA ligase demonstrated much variation between molecules when aligned, and a FASTA line on the PDB reveals the same. In this image of a DNA ligase 1 molecule and a DNA ligase 3 molecule in alignment, you can see that we have made the internal active sites easier to see by isolating them from the protein. The misalignment, although slight, would assist in creating a differentiating factor between 2 molecules, allowing for further prediction capabilities. To find the center of our DNA molecule, we first graphed the phosphorus molecule specific to the DNA backbone. From there, we ran a plane of best fit function, from which the center can be used as our reference point. We are also working on curve fitting the DNA and finding the center of the curve in order to determine our reference. Hold on. There we go.
So now for our results. So in the past few months, we've determined which features we're going to use to classify the DNA ligase molecules. And these features are based on the relative geometry of the DNA ligase molecules. So to determine the differences in the geometric features of the molecules, we aligned and analyzed the DNA ligase 1, 3, and 4 in PyMol, as you can see in this figure. So this model shows an alignment between DNA ligase 1 and 4. And as you can see at the bottom, the alpha helices and beta ribbons are positioned differently between the 2 isoforms, which shows the geometrical difference between them. Then, in order to determine the relative geometry of any DNA ligase molecules, we plotted the DNA backbone using the coordinates of the phosphorus atoms. And then using a surface fitting function to map the central axis of the DNA backbone, we determined a function to find the center of the DNA backbone, which would then be used as a distance measurement for the relative geometry. So that's the figure below. That is a model that represents the DNA backbone, and the plane is a result of the surface fitting function.
Oops, there we go.
For our future work, we have so far determined the factors that we need to determine this heuristic for classifying DNA ligases, and we have, but we're still in the process of developing the models and methods to be able to classify and get some tangible results. And our next steps would be to finalize and refine the plane that we have created to identify the center of the molecules. And use systems such as PyMol and Chimera to determine the distance between DNA ligase molecules and the center, which will then be used for classification. And then we plan to implement a supervised machine learning model such as a KNN, K-nearest neighbors algorithm, or a neural network that would then use this data we found from the plane and provide information on whether or not a correlation exists between the relative distances and determine the type of DNA ligase. And most of our work so far has focused on classifying different types of DNA ligases, but this approach can then be used to apply and classify other types of proteins in the chemical space.
So for our conclusion, even though we have not yet conclusively determined a heuristic to classify DNA ligase molecules, our results show that DNA strands can be mapped with their phosphorus atoms. So that can help us find the coordinates of the center of the DNA strands, which, as we mentioned earlier, can help us find the distances to the relative geometry. Then by determining whether or not we can classify DNA ligase molecules based on the relative geometry, we will then be able to take further steps in our final effort to actually predict protein structure based on a 1D amino acid sequence. Since there's so many proteins in the PDB, we aim to facilitate the structure prediction process by finding similarities to existing protein structures. And so our next steps are going to be to collect the relative geometry distance data. And as Niranjana talked about, is to use machine learning to classify DNA ligase molecules based on this data.
All right, that's pretty much it. Here's the QR code to our reference page. And we thank our advisors, Mr. Downing and Mr. New, and we thank ASDRP to give us this opportunity to work together on this. Thank you.